Hello, my name is Abigail Borgmeier, and I'm presenting results of a study examining Eudorylaimus taxonomy and distribution within the dry valleys of Antarctica. Eudorylaimus is one of few nematode species present within the continent of Antarctica. It's an omnivore predator, placing it at the top of the food chain within the soil ecosystem. This nematode has a very long lifespan, particularly in the Antarctic environment where the active season is very short. And despite being one of few nematode taxa present on the continent, there are many discrepancies in how many species of Eudorylaimus are present in the Dry Valley region, as well as what's the accurate species name for the dominant taxa found. The Dry Valley region is characterized by being quite dry, except for areas where Eudorylaimus is found, particularly recently wetted soil, so near streams, lakes, or around glacial melt. Everything, or most everything we know about Eudorylaimus taxonomy is from a 2008 paper by Andrassi, and this study described 6 different Eudorylaimus species from the Dry Valley region. However, this study relied solely on morphologic characters, and there's no genetic analysis behind these species descriptions. Since a 2014 paper, Eudorylaimus antarticus is the only species name used to describe any Eudorylaimus individual found from the dry valleys. However, there has been no union between DNA and morphologic analyses to support an accurate species ID or population level differences that might lead to new species found. So the purpose of this study is to do exactly that. I sampled soil from the 4 major valleys within the larger Dry Valley region in order to try and find population level differences, as well as sample from different microhabitat types. And these types included dried algal mats as seen in the center, moss beds, as well as recently wetted soil from glacial melt. And so in order to combine both morphologic and DNA analyses, nematodes were extracted from the soil, and then single Eudorylaimus individuals were first photographed, then measured for key morphologic features. And then that same individual DNA was extracted from and then sequenced using both the 18S nuclear and the CO1 mitochondrial genes. There are several key features used in taxonomic keys based on morphologic characters. And after preliminary results, body length in particular, as well as other features, do not match Eudorylaimus antarcticus, which is the dominant name used in the literature currently. So most papers are not using the correct species name for Eudorylaimus within the dry valleys. However, DNA analysis is necessary to find if there are multiple species present or if a species redescription is necessary in order for taxonomic ID to be accurate. This is a phylogenetic tree built using maximum likelihood, and the different geographic locations of these Eudorylaimus individuals are highlighted in blue. And these are CO1 mitochondrial genes. So this tree shows that the CO1 mitochondrial gene for Eudorylaimus, even from geographically distant places, is nearly identical. And this supports that there's only one species present in the dry valleys. However, the morphologic characters show that it is not being correctly identified. So despite the Eudorylaimus being one of few nematode taxa, the accurate species name is not being used currently. And so further genetic and morphologic analysis are necessary in order to determine if a species redescription is necessary, or if a different species name should be used in order to correctly identify this nematode from the Dry Valley region. Thank you.